biology video, and today we are going through 6.1, which is homeostasis, and we start off with homeostasis and hormones. I've put the title hormones as more homeostasis, but I think hormones is quite important to introduce. So what we need to know to start off with is that homeostasis is the maintenance of a constant internal environment. So nothing different from GCSE, apart from the fact that you need to obviously be aware that it doesn't matter what the external environment is. So here we've got a picture of what looks like a pigeon, okay, and we're saying that the external environment, they've got large fluctuations, let's say of temperature, for example. The temperature is going up and down, up and down, up and down. However, inside, but whilst the external environment is changing, the internal environment is hardly changing. It's staying in between a very small range, effectively. So the internal environment does change because if you think about humans, our human body temperature, the optimal temperature is 37.4. Rarely during the day will you be at 37.4, but you are at an acceptable range, say between 36 and 38 degrees. You rarely go up to 41 degrees, which wouldn't be acceptable. So homeostasis is the maintenance of a constant internal environment within an acceptable range. So there's a dynamic equilibrium fluctuating around an acceptable range. Okay, so this is all controlled by something called a negative feedback loop. And basically negative feedback is a mechanism which restores the levels in our body to the original level it should be, or reverses a change in our body. So for example, if we look at temperature control, if we thermoregulation being part of homeostasis, if our temperature increases above 37 degrees, what will happen is sense organs, so sensors in the skin cells and the thermoregulatory center in the hypothalamus in the brain, will detect the body temperature has increased. It will then coordinate that response, and it will tell our effectors, and if you remember from GCSE, effectors are always glands or muscles. It will tell the effectors to change something. So for example, the effector in this case is a sweat gland, and the sweat glands will release sweat, and then that will cause the body temperature to drop, and it will reverse the change. Okay, so the effectors are what causes that change, but it's a negative feedback loop, whereas a mechanism that reverses a change in our body. Now there are examples of something called positive feedback. So I wrote that wrong, so apologies about that. That should say positive feedback there. Okay, so positive feedback is a mechanism which further increases the level away from the norm. And there are some examples where this is good. So for example, if you cut yourself and there's a tear in a blood vessel wall, what happens is clotting, obviously, from platelets, okay, and the platelets stick to chemicals, to the site, sorry, and release chemicals. Those chemicals then continue to attract more platelets, which will then cause more chemicals to be released, which will attract more chemicals and more platelets because more chemicals will be released. So that's an example of positive feedback. So there's more and more platelets can be releasing more and more chemicals, which attracts even more platelets. And obviously, once the cut initially is actually healed, then the platelets will stop attaching and the chemicals will stop being released. Another example is during childbirth. Okay, so in childbirth, you get, if we start from here, so we'll say that the baby's head pushes against the cervix. Okay, that causes nerve impulses, which transmit to the brain. Those nerve impulses cause the pituitary gland to release a hormone called oxytocin. Again, oxytocin is in males and females, but in females, oxytocin is involved in pregnancy and giving birth. Oxytocin is then carried to the bloodstream to the uterus, and oxytocin triggers contractions, okay, which pushes the baby's head further into the cervix, which then triggers more oxytocin to be released, and so on and so on. And this negative feedback continues as the contractions get higher and higher and higher until obviously the contractions push the baby out of the uterus and the baby's born. So positive feedback can be useful. It can also, however, be not so useful. So whilst negative feedback was used to control temperature, positive feedback can happen when the homeostatic systems break down. So again, should have wrote positive there, I do apologize. Okay, and what might happen here is, for example, in thermoregulation, hyperthermic assessment. So an example might be of positive feedback is that if you are really cold, okay, one of the lot of the symptoms, you get clumsiness, slurred speech, confusion, and that is because you're getting colder. Your enzymes are starting to work less and less. Now, because your enzymes work less, that means that your brain starts to function less, and because your brain starts to function less, that means it stops controlling your temperature and will stop fighting against the temperature drop, and therefore it will allow the temperature drop to continue. So that's obviously a bad example, as a negative, not very good for us, positive feedback example. We've then got hormonal communication. So this is a key bit to introduce to you. You need to know what a definition of a hormone is. Hormones are normally proteins, but there are some examples of lipid versions. So a hormone is a chemical messenger produced by an endocrine gland and transported into the blood to a target organ where it attaches to a receptor protein and it activates an enzyme inside a cell. Now, that is if it was a protein. Lipid-soluble hormones will just diffuse straight into the cell and combine with a receptor in the actual cytoplasm, but the protein hormones can't, so they have to attach to receptor sites on the outside of the cell surface or plasma membrane. Now, this is obviously all to do with hormones, so you need to know those definitions. So again, we need to know hormones are chemicals that transfer information from one part of the body to another, a lot longer lasting and slower than nerve impulses. Endocrine, you'll be doing this in the next lesson, but an endocrine is anything that releases a hormone, so an endocrine gland, whereas exocrine releases substances that are not hormones into the gut, into ducts, like the gut, for example. So exocrine could be enzymes, whereas endocrine glands could be hormones, and they always go into the blood. Okay, and you've got the following endocrine glands, which you should know from GCSE. So you've got the pituitary gland, you don't need to know the pineal gland, the thyroid gland, which controls growth, the thymus, you've got the adrenal glands on the kidneys, you've got the pancreas, which we're going to talk about next lesson, you've got the ovaries and the testes as reproductive glands. Now, in terms of being ready for action, most endocrine glands can secrete hormones very, very quickly. Adrenal glands are obviously the quickest, obviously for their reason. And a lot of hormones will have a short lifespan or half-life, as they'll be broken down in enzymes. Insulin, for example, is 15 minutes. Adrenaline is about 3 minutes. But obviously, it's longer lasting than a nerve impulse. Now, how most hormones work, we've already sort of said about this, but you need to know this in quite a bit of detail. So a protein or a peptide hormone, which includes insulin and includes adrenaline and things like that, cannot pass through the phospholipid bilayer of a membrane. So we have something called a second messenger system. Okay, so whereas steroid hormones, which include sex hormones, so estrogen and testosterone, they can pass directly through, we need to know about this second messenger hormone. So what we have here is the example of adrenaline, which is a amino acid hormone or a protein hormone, and that is what we call the first messenger. So it takes the message to the receptor on the outside of the target cell that is on the cell surface membrane, and there's a receptor site on the cell surface membrane. That then causes, once it's attached, it's got a complementary shape to the receptor. That then activates an enzyme inside the cell surface membrane. And in this case, in the example you're expected to know, is adenyl cyclase. And as a result, then what happens is adenyl cyclase converts or becomes active, okay, and that converts ATP into cyclic AMP, okay, and cyclic AMP is a example of a second messenger which then activates the relevant enzymes inside the cell. So, for example, in this case, it might be something that activates the production of glucose in the liver cell to increase your energy available from respiration. So again, you need to know the primary and secondary messenger. The first messenger is the hormone, the protein hormone, which attaches to a complementary receptor site that activates adenyl cyclase, and then that converts ATP into cyclic AMP. And it is cyclic AMP that is the second messenger that activates the enzymes that causes the cell's response to the hormone. So you would be expected to answer a question like this. So explain how adrenaline triggers a cellular response. So pretty much what I talked you through there. So you'd be expected to say that the adrenaline is the first messenger. It is a protein hormone, so it can't go through the phospholipid bilayer. It attaches to a receptor cell on the plasma membrane that has a complementary shape. When it attaches to the membrane and calls a complementary shape, it activates the enzyme adenyl cyclase, and adenyl cyclase converts ATP into cyclic AMP, and then cyclic AMP activates the enzymes that are involved. And in this case, it is an enzyme called protein kinase that converts glycogen into glucose phosphate, which could then be further converted into glucose. And that would be how glycolysis works. Okay, and this is all an example of negative feedback. So remember what negative feedback is. There is a change and it's your body reversing a change as a result. So for example, the receptor detects a change, okay, then the body it goes back to the normal temperature of the body, and that's what it does as a result, okay. So factor might start off at a normal level, and then if the temperature increases, the receptor detects that, and then the effector takes it back down to normal. If the temperature goes too low, the hypothalamus detects it in the brain, and it takes the temperature back up again. So I hope that's been useful, guys, and I'll see you soon.